All right, well, welcome to the Opera House. Thank you for coming this evening. It's my pleasure to welcome you here. My name is Ben Twining, and I am the Vice President for Education at the laboratory, and will serve as the MC for the evening. I want to start by thanking the Opera House for letting us host this here. Some of you might know we are building some new buildings at Bigelow, and look forward to welcoming you all back to our campus next summer. So, Bigelow Laboratory, as you probably all know, is a laboratory that focuses on microbial oceanography and the life at the base of the food web. And as a nonprofit research institute, the discoveries, solutions, and inspiration that we create are made possible by the help of many generous supporters. And so I want to start today by thanking all of the board members, donors, and advisory board members who make our work possible. So thank you. Truly appreciated. I also want to extend a special thanks to H.M. Payson, a Maine-based financial advisor who has supported this series for many years. So many thanks to H.M. Payson as well. And now it is my pleasure to introduce our speaker tonight, Dr. Nicole Poulton. Nicole came to us back in 2001 as a postdoc. You're going to see some pictures of that auspicious moment. Prior to coming to Bigelow, Nicole got her undergraduate degree from Virginia Tech, and then did her PhD work at Woods Hole Oceanographic in the MIT Joint Program, and then came to Bigelow as a postdoc. And Nicole, in addition to being an amazing phytoplankton ecologist, I have to mention, is a champion and leader of some of our great education programs as well. She leads the high school, yes, we're here. She leads the long-running Bloom program, Keller Bloom program for high school students, and also our Bloom Educators program, and is involved in lots of education. So, a true star. So with no further ado, Nicole, hand it to you.
All right. Can everyone hear me in the back? Good? Yes, wonderful. I just want to make sure that's loud enough. So, microscopic wonders of a drop of seawater. But before we get to that, this is our 50th anniversary at Bigelow Laboratory. Pretty amazing. So, a little background about myself, although you had a great introduction from Ben Twining. In 1974, I was on a beach building sandcastles on the coast of Lake Michigan. So I grew up in the Midwest, in Michigan and Indiana. I was born in Indiana. And then had the opportunity, and so to really get the bug or the interest of being working on the ocean is really when I moved to the South Pacific as a kid, as a teenager. So my father was the director of planning and development of the University of the South Pacific. And so I had this unique opportunity and first time really to grab a hold of how big the ocean actually is. So today I'm going to be talking to you about how I got my love of microbes, and we're going to be talking about a new tool, not a new tool, but it's been around for about 30 years now, 30 plus years. And this is flow cytometry, and it was developed within the biomedical community, and I'm going to be talking to you about how it was pioneered here at Bigelow Laboratory for use in ocean sciences. It's really revolutionary in how it's really revolutionized our research. So as Ben just said, I arrived at Bigelow in 2001, 23 years ago, and phytoplankton are near and dear to my heart. And I've been working in the science of the Gulf of Maine for quite some time, and now I'm branching out to a lot more global science. And as I mentioned, I direct the Center for Aquatic Cytometry now at Bigelow Laboratory. So let's get back to that drop of seawater. So if you were to take a single drop of seawater and put it onto a microscope slide, you would see that it's teeming with life. So we have phytoplankton in this image here. This is a micrograph from a microscope, and they range in size. There's some that are very large, some are very small, and they're really no bigger than the width of your hair. So some of them are actually 100th the size of the width of your hair. So they come in many different shapes and sizes, as I've mentioned. And they're globally very important, and this is for all microbes. So the challenge is in this soup of microbes that are out there within that drop of seawater, how do we visualize and study these organisms? And we have many tools, some that have been around for centuries, such as microscopy. And you just saw a whole bunch of micrographs there that have been used. And we still use that tool today a lot. So flow cytometry, however, is a new tool. So within the last 30 to 40 years, and we are now using that tool to understand the distribution of these organisms and study them. But we can also study them genetically, and you will learn next week from Dr. Ramona Stepanauskas how you can actually study the genomes of these different microbes individually. And then the other thing that we do really well at Bigelow Laboratory is that we have a culture center where we've been cultivating these organisms for the last 50 years and really allowing researchers to study these organisms in greater detail. So, how many are there out there in that drop of seawater? So, let's take a little deeper look into here. So if you were actually to go even further than phytoplankton and look down to the smallest forms of life that we have in that drop of seawater, we're talking about 50,000 to 500,000 bacteria and viruses on average, so about a 1-to-10 ratio. But if you were to step back to the organisms that I like to study, more so than others, is phytoplankton range from about 500 to about 5,000 per drop of seawater. And again, they vary in size quite a bit, so that range is sort of indicative of the size of the organism. And then you get very small zooplankton. These are the predators of the bacteria and all that phytoplankton that's in that drop of seawater. And that ranges from about 1 to 5. So, as I mentioned, microscopy is the tool that we've been using for centuries. And here's an image of Wendy Bellows, who was a research associate, and she was with us for many, many years at Bigelow Laboratory, now retired. But she used traditionally a microscope to count these organisms, as does Peter Countway, as a current senior research scientist. This is during one of our culture courses at Bigelow Laboratory. And myself, as a postdoc, back in the day, taking images of these organisms under the microscope. Something that these organisms have that we use quite a bit to help discern these organisms from other organisms is that they have pigments, and that's that discoloration that you see. And you can see here the brownness that's there, and some of them have lighter and darker amounts of this pigment. Anyone want to know what that pigment is called? The predominant one is chlorophyll. And as you can see here, these are many different strains that we have part of our culture collection at Bigelow Laboratory, and they range in color. So there's many different pigments that are there that can be used to help discern these different phytoplankton. So here's a bright field micrograph of some phytoplankton. However, if we were to shine a very specific wavelength of light, such as blue light, on this organism, or these organisms, it would actually return light. It would absorb some of that light and then return some of that. And so these pigments are fluorescent. So it allows us to visualize these organisms in a different way using microscopy. So this is what we call fluorescent microscopy. We can use different wavelengths of light and look at different types of fluorescence. In this case, we're looking at another pigment. In this case, it's called phycoerythrin, but it's within the same organism. So this organism has 2 different pigments that we can discern. And lastly, from the biomedical industry, we have learned that we can actually probe or tag these organisms using a DNA stain or probe, so they can attach their DNA. And you can actually color code and quantify the amount of DNA using these fluorescent techniques. So for chlorophyll detection and DNA detection, we can now zoom in and start to look at these other microbes, such as bacteria and viruses. So what we have here is a micrograph of bacteria and viruses. We're zooming in at much higher magnification now. And all of these have a DNA probe that have been applied. That's the only way we can really discern them using microscopy. Now, would anyone like to count all the bacteria that are on the screen? So you can probably imagine that this would take some time to do this. So we visualize this with fluorescent microscopy, but if we were to zoom in, the bacteria are these larger of these these green dots. And if you really zoomed in a little bit further, you can actually start to see the little tiny dots in the background. Those are the actual viruses that are in a drop of seawater. So counting these cells is very challenging and a very slow and tedious process. So what is one of the other ways that we can do this? So from the biomedical industry, flow cytometry is a very rapid method of counting and categorizing these cells. And we do this using the laser light, actually. So instead of shining a light down through a microscope objective, we are actually using laser light to discern how big the particle is based on its scatter, and then how much fluorescence it has. So cytometry means cell measurement. So this is typically done on cells, but it could be done on many different particles. As I mentioned, the biomedical applications, we have lots of environmental applications, and there's lots of different instruments now in the current state-of-the-art flow cytometry labs where you can have very different capabilities. So the biomedical industry developed this tool back in the 1960s and 70s. And this is one of the larger. This is a Coulter Epix 5 here. And it's been used primarily and currently used as used in cancer research as well as a pathogen diagnostic tool. So traditionally, in the past, it was actually used to diagnose HIV and AIDS in people and to help diagnose and understand blood cancers, and still does. And even today, immunotherapies are being used to isolate antibodies against, for example, the COVID virus that we've been experiencing since the pandemic. So what does a flow cytometry look like, and how do we analyze this data? So this is a video that shows how the principles of a flow cytometer works. So we have a sample, our drop of seawater, and we're putting it through the instrument, and those particles, each that you see flowing through there, represent microbes in the ocean, in this case. And we've tagged those microbes in different ways, or they have that natural fluorescence, which is exhibited by chlorophyll. And what we're seeing here is individual particles passing in front of the laser beam. And every time a particle passes in front of that laser beam, we're gathering information, and we're doing it at rapid speed. So what we have there is we have the size of the different individual molecules that are in that sample, as well as that fluorescence that we can measure. So we can measure how many phytoplankton are in the water column, or maybe how many bacteria are in that sample. So what are the advantages of this technique? As I just said, we can now quantify these different particles at rates that we could never do using microscopy. So we now have particles that we can now look at 100 or 1,000 times per second or 100,000 times per second. And as I said, this really reduces your analysis time. Here's a flow cytometer that's currently in our laboratory, and you can sort of see where the laser beam is here, and there's a fluid stream that's coming down in front of that laser beam. The sample is coming in from an ocean sample into here in front of this laser. So what we're seeing is that we're not getting images from a flow cytometer or a traditional flow cytometer, but what we're gathering is how big the particles are and how fluorescent they are. So for every dot that you see on that graph represents an organism. It could be a phytoplankton. In this case, we're looking at the pigment. If we're looking at chlorophyll, these could be all phytoplankton, or we could be looking at bacteria and viruses and quantifying them very rapidly. What's another advantage of flow cytometry? A different tool called the cell sorter could be used to isolate cells of interest from the water column. So in this case, we have our traditional flow cytometer here with a laser beam. These cells are then tagged based on their fluorescent signature. Oops, I can't go back. And so what we're having here, we can establish cultures. We can divert the sorted particle to the side. These are droplets that are formed of individual particles that might be in there. And we can do downstream analysis on them, for example. So next week, when if you were to return to see Dr. Ramona Stepanowska's talk, he's going to talk about how we can isolate single cells from the ocean and basically get their whole genome from a single cell. So now I'm just going to step back in time and talk about the pioneering vision that Bigelow had here in Booth Bay. So we have our founder, or one of our founders, who's also here in the audience, Clarice Jentsch. And this was back when the biomedical industry was really just developing this tool. And now they were trying to think, well, maybe we could apply this to ocean sciences. And so back in the early 1980s, Clarice received the first funding to purchase a flow cytometer at Bigelow Laboratory. And from there, this is Terry Cucci, who was a flow cytometrist working with Clarice. And this is the first flow cytometer that was purchased, similar to the one I showed you previously. This is a Coulter Epix 5. And in 1983, the flow cytometry sorting and Natural Resource Center was established and was later called the J.J. McIsaac Aquatic Cytometry Facility. So this was the first time we could start to use flow cytometry to study organisms in the ocean. But we wanted to spread the word about that. So one of the things that Clarice first started to do was bring scientists together to see and understand the potential of flow cytometry at Bigelow Laboratory. So what she did over the years, starting back here, the very first workshop in 1983, and going forward through time, we've had many, many workshops trying to introduce flow cytometry to different scientists and understanding the potential of this science. And she had our first one here in West Booth Bay Harbor at our old location, where the DMR location, or the Department of Marine Resources, is currently located. And this was the first course that took place at West Booth Bay Harbor. And since then, we have participated in many. I've taught quite a few. This is in 2017. And there's a few people who are currently at our laboratory here. And even today, in April of this year, we actually held the course. It had been a few years because of the pandemic, but we finally got back here. And this is a photo that we took. We did a field trip out down to the boat, and this is our new Bigelow research vessel where we house another flow cytometer. So with Clarice's vision, she had the idea, oops, sorry, I went backward. I'll leave that up there. So she had the vision for going to the Society of Analytical Cytology and talking about the potential of cytometry in aquatic sciences. And from this, there was a special issue that came out that brought together many scientists within the discipline. This was in 1989. And what it showed us was that phytoplankton detection could be taken place. We could look at invertebrate grazing of phytoplankton. We could use these instruments at sea and onboard ships. And we even developed preservation protocols. How do we bring back samples from the ocean to run on these flow cytometers? And there are many, many people that were instrumental in the establishment of this initial center here at Bigelow. And that's Clarice Jentsch, we have David Finney, Terry Cucci, Ellen Hogan, and my mentor and following director, Michael Siraki. So now, let's just talk briefly about a discovery that came out about the same time as this special issue that Clarice was working on. This discovery in flow cytometry was the discovery of a small cyanobacterium called Prochlorococcus. And in this field of view, going back to our bacteria sample, you'll notice that there is a colored cell here in the center here. That is a type of cyanobacterium. And you can sort of see how difficult it is to discern that from other particles in the ocean. But it was because of the increased sensitivity of a flow cytometer that we were able to identify this organism. And Penny Chisholm, I'll bring her up here, this is one of my former mentors. As an undergraduate, I had the opportunity to work in her lab, and I also was I'm a research technician for her for a year. And this is where I learned flow cytometry for the first time 25 years ago. And Prochlorococcus was what we called this organism, or what she identified this organism as, and it was the most abundant photosynthetic organism in the world. And how did we determine that? Through flow cytometry. And this was a paper that was published in 1988. So since then, we've looked at the distribution of this organism across the globe, And here are sampling locations that have been taking place, across the globe. Each one of these red dots that are on here have been run, these samples have been run through a flow cytometer to get an idea of how distributed this organism is in the water column. Well, actually around the globe. So this is surface concentration, and what you're looking at, it spans, this organism spans from the 40 degrees south to 40 degrees north, and you can see there's areas that have huge abundance over probably 1 million per milliliter. So since then, scientists have been modeling this organism because it's so critical in our global biogeochemical cycles, so much so that you can see they've been modeling everything that has Prochlorococcus is in this green color from dark to light. And you can sort of see where it spans across the globe here. And there's 3 other types of, or species of phytoplankton also distinguished here, especially diatoms, which are usually found in our temperate regions of the planet. But this Prochlorococcus, without cytometry, would not know how important it is. It's really quite amazing. There's a billion potential Prochlorococci out there, estimated. So that is 10 to the 27th. That's a lot, but it's 20% of the oxygen on the planet. Every 5th breath that you take comes from this organism. So very, very important. So I'm going to go back to my timeline now. That was a little vignette of what was happening at the same time Clarice was publishing these special issues and the importance that these organisms in general were having using flow cytometry. So we have here my mentor, Mike Siraki. Clarice Jentsch moved on to other opportunities. She left Bigelow Laboratory, and Michael came in as the director of the center at that point. And that's a time when there was this innovation that was taking place, an idea for imaging flow cytometry, combining both flow cytometry and imaging flow cytometry into one. And doing that would generate, what if we could take an image for every particle that passed in front of this instrument? We would have the flow cytometer and microscope. And this was a co-invention between Chris Siraki, who is also here in the audience, Mike Siraki and Charlie Jentsch, one of our founders at Bigelow Laboratory. The innovation really allowed for basically particles to pass in front of a laser beam, but then also be captured on the camera so the particles are being visualized through a flow cell here. This allowed for the publication in 1998 of imaging flow systems for automated analysis in marine microplankton. It's the first imaging flow cytometer on the market and developed anywhere. So this just gives you a sense of how fast images can be captured by this instrument. The particles are flowing from the top to the bottom here, and there's a laser beam that's capturing these, that's actually triggering the camera to take an image, and they're being collected over here. The potential here is huge. As you can see, this instrument can basically allow for the automation of microplankton detection. And the innovation over time, this company, now Chris Siraki, went off to found the company called Fluid Imaging Technologies, and it's now celebrating its 25th anniversary. And speaking directly out of Chris's mouth here, flow can revolutionize a tedious and slow process of manual examination of phytoplankton via microscope by providing a semi-automated method to rapidly count, measure, and analyze individual cells and particles in fluid using digital images. And that's our founder. So, where did I come in this? So, in 2001, after the innovation of imaging flow cytometry, I came into the world here at Bigelow Laboratory as a postdoc, bright-eyed and bushy-tailed. And these are some of the key players that I've had over the years to work with. And I have been working using flow cytometry and imaging cytometry for quite some time, and I'm going to tell you a little bit about that research now. And to do that, before I did that, in 2014, after Mike Siraki unfortunately left the lab, I became director and now direct the Center for Aquatic Cytometry. We have brought to the laboratory now many instruments. We have over 5 new instruments at the laboratory over the past decade. And what that's allowed is that we're a service center. We provide the aquatic science community an opportunity to do science at Bigelow because these instruments are expensive. In the biomedical world, they can afford them. I mean, they replace them every 5 years. Here, I've been keeping these instruments going for 10, sometimes 20 years that we have to keep these instruments going. So, my research, how are we using these flow and imaging tools to understand a changing ocean? So to do this, I'm looking at just one aspect of my research, which is right here in Booth Bay. And we have a time series that Michael Siraki, one of our former directors, started back in 2001 when I started. And it's right here off the McCown Point dock, if some people may recognize this. This is where Bigelow was founded. This is where Bigelow began at the beginning of our 50-year history. And now it's the Department for Marine Resources. But this dock right here, I've been sampling on a weekly or every 2-week basis since 2001, looking at the phytoplankton and the bacteria within the samples there. And this is Booth Bay time series. I just wanted to, quick note, the water mass is actually called Booth Bay. A lot of people don't know that. All right, so changing Gulf of Maine. This is the Gulf of Maine right here, and this is where the Booth Bay time series is currently located. And as you can see here, this is an anomaly plot of temperature. So going back to 1982, we're basically seeing each one of these is a year, each row is a year here, and basically where you see warmer or redder temperatures, that's where a warming is occurring. There's a positive anomaly in the temperature based on the average of the entire data set. And this is where there's cooling happening. But you can see here, literally, for the past 15 years, we've been seeing warming taking place here in the Gulf of Maine. And the time series really situates itself quite nicely across that location, across those years. So one thing that we've been monitoring using traditional flow cytometry is the abundance of these small phytoplankton. And we've been looking at their distribution over time and how they've changed. And what we're seeing is that there's an increase in their abundance over time of these smaller phytoplankton. And we're also seeing potential, what we call phenology shifts, timing in these blooms. You can sort of see over time that there's getting more dense, and here is basically there's more throughout the year. These are our abundances that are mean abundances from January all the way to December, looking over time, beginning of the time series to present day. This is actually 2020. I've not updated it yet. But what's really been interesting, how do we know that there's increasing abundances? Well, we can look at this anomaly plot. And what we're seeing here is there's a positive change in the change in these distributions. So why is this important? This is really important to understand how different organisms potentially, having smaller organisms or smaller phytoplankton and maybe less larger phytoplankton could change how things are being moved through the food web, especially up into fish, for example, and are feeding our fish. Now I'm going to quickly, before I end this portion of the talk, I wanted to quickly mention monitoring phytoplankton dynamics from the same time series. What we're doing here in Booth Bay, we're using an imaging cytometer in this case. These are images. These are the black and white version of the image or the instrument that I have. And we've noticed that there's a specific organism in here that we can pull out. This is what we call mesodinium. It's a prey organism for another organism I'm going to point out. And that is Dinophysis, which is a predator. And so what we can do is bundle these different organisms together and look at their distribution over time. And that's what's so great about a time series. You can see how these two organisms are interplaying with one another. So what we see here is that we have mesodinium, and I'm looking over 4 years, 2008 to 2011. This is from January to January, so this is one full year. And you can basically see this is a running average of distribution of abundance of mesodinium over time. Notice that there's a peak drop-off throughout each year. And then when you look at the predator, which is feeding on this population, you can sort of see where it dominates every year. Basically, where there's a fall in the prey species, you're seeing an increase in the predator. And this specific organism is a toxic species, or potentially toxic species. So it's an important one for us to understand and monitor, because if there's phenology shifts that are happening potentially due to climate change or warmings of our oceans, then we'll need to understand these dynamics in the past and seeing how they're changing into the future. So I'm going to end this section right now and probably take a few questions about, basically we are understanding that this area here around us is rapidly changing, the Gulf of Maine. And understanding the phytoplankton phenology, abundance, distribution, and different composition is really important to understand the shiftings and potentially changing food web interactions that are out there. So with that, I'm going to pause for a few minutes because then I'm going to take a look forward. So how can we take what we're learning here from Booth Bay into the future? All right, so celebrating the past and solving the future. So now we're going to take a look forward. I'm going to start just a few years out, but maybe how can this help us 50 years out? So how are we going to use these tools to understand changing ocean and human impacts? So getting at your question down here in the front row, one of the things we are working on are human impacts is detecting microplastics. So, and I did not plant this. So what we have here is the ability to characterize and isolate different microplastics using this tool, flow cytometry. So we've been able to do this because I have found, or actually it's in the published literature, is that you can adsorb one of those fluorescent stains or probes to the surface of microplastics and make them fluoresce. This is a fluorescent microscopy image here. And you can actually then identify these different groups not identify them, but what you can do is sort them. Remember I talked about the cell sorter and isolating those different organisms? Sorry, I keep saying organisms, particles. So you can actually isolate these microplastics and discern what type it is. So for example, is it PVC? Is it, I'm blanking all the different names right at the moment. So basically there's different types of different plastics that we can actually quantify and also identify using downstream chemical analyses. So that is one future outlook that I plan to use with flow cytometry because it's not only able to look at cells, it can look at particles in these different environments. All right, so how will plankton communities change over the next 50 years? So speaking to another question in the room earlier, what we're seeing in our warming oceans is that there's a change in potentially I'm thinking from a phytoplankton perspective, being a phytoplankton ecologist. So what we're having here is that traditionally there is mixing taking place at the surface, and that's bringing nutrients from the deeper ocean to the surface that is feeding our phytoplankton. However, in a warming environment, what's happening is that the surface layer is getting much more warmer, and it's forming a barrier between the surface and the deeper water. And so there's less nutrient flux to the surface. So what's happening there is that you can sort of see by the size of my phytoplankton, there's less nutrients, so basically there'll be smaller phytoplankton that will exist usually in that environment. And so that is just one example. But in a sense, how are we going to be able to monitor this on a global scale and look at this? And this is a plug for one of our talks in 2 weeks, whereby we're going to do this using satellites. So, and when we do that using satellites, we had just launched a new satellite into space in February of 2024, earlier this year. It's called the Plankton Aerosol Cloud Ecosystem Ocean Color Instrument. We just call it PACE. And what this instrument does is it's going to be looking at the surface ocean of the planet pretty much on a 24 to 48 hour time scale. So what it will be able to do is map where chlorophyll is located, that pigment I talked to you about with flow cytometry, and also the distribution of particular organisms. Pico-eukaryotes, pico means small. Those are small phytoplankton that we're looking at. Synechococcus is one of those cyanobacteria that I was talking to you about. This is on a coastal system, so that's why we're not seeing that wonderful Prochlorococcus. But what we're seeing here, this is the southern South African coast, but this is just one example of how we could potentially monitor from space. But where does flow cytometry come into all this? I was recently funded by NASA to use flow cytometry to validate the satellite data. An instrument is only as good as its calibration. So the only way to make sure the satellite is registering the returning light to the satellite is to use actual flow cytometric measurements from in the water systems that it's observing. So what we're doing here, we're going to sea with ships, and I have on board here, this is a newer spectral flow cytometer. I won't go into all those details, but it's very similar to the way the satellite is detecting images from space. And so what we're having here is we actually have water that's being pumped in from the side of the ship. It's actually coming right in here into the side of the ship. And basically it's being pumped right into the flow cytometer. And this is done we're taking a sample every 20 minutes while the ship is moving. So high resolution, lots of data. This is something that hasn't really been done before, and we'll be able to doing it at high resolution. So we'll be able to sort of assess how fast that change is happening in the ocean globally. So how can we continuously monitor plankton in a changing ocean? Let's see. Can we classify plankton in real time? This is getting at another person's question. Is it possible? So we're generating a ton of this image data. So one way of doing this is to how to monitor continuously in the water is can we deploy our flow cytometers into the water? And there are now instruments or tools. This is the imaging flow cytobot. They get really creative with their names. Or CytoSense, which is made by CytoBuoy. So anyway, point side, but these are submersible flow cytometers. These can go into the water column. And they have imaging instruments that have been deployed at sea, and they can be there for a long period of time. However, they require a lot of energy to function. So one thing I was thinking, and this is actually being done by a colleague of mine, is that how can we power these instruments remotely, and how can they actually be out there mapping the distribution of phytoplankton or other microbes? Well, we're becoming a lot better at designing unmanned vehicles such as autonomous boats or drifters. And this is a fairly new concept, and this is made by a company called SeaTrack. And what we could do is actually deploy a flow cytometer from an autonomous boat. And it could drift around, or not just drift, actually, it actually propels. It actually has a propeller here on the back. So you could actually send it on sort of a trajectory out there in the ocean. This is taking place now in the Gulf of Maine to monitor for harmful algal blooms now. So, and we're using an imaging flow cytobot, which I mentioned in the previous slide, to do that. That's not done by me, but what I hope to do, I just literally 2 weeks ago got my first imaging flow cytobot. So I'm very excited to use that over the next decade. Can we identify and classify these species of phytoplankton rapidly? So how can we use artificial intelligence or classification algorithms? Can they help us, assist us in this way? As I mentioned, there's a plethora of data that we're going to be capturing. You saw how fast the images can be captured by these instruments. So the continuous monitoring, continuous data streams. So what we're trying to do is potentially, if an iPhone can detect who's in the audience, we can do this with phytoplankton. So it's just a matter of putting the right resources towards that. And I do believe in the next 50 years that we will be able to do that. So again, in real time, these instruments will be collecting data for us, analyzing it for us, and spitting back the data to us in real time so that we can understand the distribution, the composition of the phytoplankton, and assess the changes in different ecosystems. And with that, I'm going to say thank you to all the different people that have made this completely possible for me for the last 23 years. Special thanks to Mike Siraki, a former director here. Special thanks to Clarice and Charlie Yench. Clarice, thank you so much. And Penny Chisholm, discoverer of Prochlorococcus. I love being in her lab. She was definitely my first mentor. And I have to also thank many, many, many people that have been in and out of the Center for Aquatic Cytometry over the years, especially helping out with this time series. Laura Lubelzik, Terry Cucci, Edward Thier, Brian Thompson, Ellen Hogan, and Doug Finney. So with that, I conclude, and I will take any more questions. Thank you so much.